So we left off with the last PowerPoint. Sorry, I got cut off there a little bit. Just talking about the different blood groups. We also have plyotrophy, which occurs when one gene influences multiple characters. And sickle cell disease is a human example of plyotrophy. This disease affects the type of hemoglobin produced and the shape of red blood cells. It can cause anemia and organ damage. Sickle cell and non-sickle alleles are co-dominant, and carriers of sickle cell disease have increased resistance to malaria. And this is just to look at sickle cells, kind of the jagged edges amongst normal red blood cells. And this is a look at how an individual can carry the sickle cell gene, but just be a carrier for it. Many characters result from polygenic inheritance, in which a single phenotypic character results from the additive effects of two or more genes on a single phenotypic character, and human height is an example of that. All right, so we're going to jump ahead a little bit. The environment can also affect many characters. Many traits can be affected in varying disease by both genetic and environmental factors. For example, the effect of genes and sun exposure on the skin of one of this book's authors and his family. Here's a look at varying phenotypes due to environmental factors in genetically identical twins, as displayed by the daughters of one of your textbook's authors. And now we'll look at a little bit about the chromosomal basis for inheritance. The chromosome theory of inheritance states that genes occupy specific positions on chromosomes, and chromosomes undergo segregation and independent assortment during meiosis. And Mendel's laws correlated with chromosome separation in meiosis. So here we have a look at the chromosomal basis of Mendel's laws, and just showing how the chromosomes will separate independent of each other to get the different generations. Here's a look at the chromosomal basis of Mendel's plants laws in a Punnett square, and showing the probabilities of how many plants that you will get being yellow round, yellow wrinkled, or green wrinkled. Bateson and Punnett studied plants that did not show this normal ratio in the second generation, and what they found was an example of what we call linked genes, which are located close together on the same chromosome and tend to be inherited together. So here we have a look at an experiment revealing linked genes in the sweet pea, showing how the experiment involves crossing of genotypes, and then you can see the resulting observed production and the prediction. So it's similar, but not totally the same. So crossing over between homologous chromosomes produces new combinations of alleles in gametes. Linked genes can be separated by crossing over, forming recombinant gametes. And the percentage of recombinant offspring among the total is what we call the recombination frequency. And these linked genes can be separated by crossing over, forming what we call recombinant gametes. And the percentage of recombinant offspring among the total is what we call the recombination frequency. So this, again, is just showing how the production of recombinant gametes. So this just means that when you have homologous chromosomes, they can cross over and produce differences in the parental and recombinant gametes. And this is just an example of this in the Drosophila melanogaster, which is a type of fruit fly. So this is a fruit fly experiment demonstrating the role of crossing over in inheritance. So we cross over a gray body, long wings, the wild type, with a black body with vestigial wings. And these are the parental phenotypes and the recombinant phenotypes that we get. And this explains how the genes in the loci will cross over. So here's a look at the experiment with the fruit flies and the differences in the offspring, and then the explanation of the homologous chromosomes and how they cross over. I won't ask you guys too many specifics about this, but you should understand how crossing over among the chromosomes does produce new combinations of alleles. Recombination frequencies can be used to map the relative positions of genes on chromosomes, and a genetic map is just an ordered list of the genetic positions along a chromosome. And a genetic map based on recombinant frequencies is called a linkage map. And this is just a look at how we can map genes from crossover data showing how the different genes will have different spots on the genetic chromosome. So the last part in this is we'll talk a little bit about sex chromosomes and sex-linked genes. So in mammals, a male has an X and Y sex chromosome and a female has XX. The Y chromosome has genes for the development of testes, whereas an absence of the Y allows ovaries to develop. And in addition, human males and females both have 44 what we call autosomes, which are non-sex chromosomes. And many non-mammalian species have other chromosomal species that will determine the sex. In some animals, environmental temperature determines the sex. So for example, for some reptiles, the temperature at which eggs are incubated during a specific period of embryonic development determines whether that embryo will develop into a male or female. And global climate change may therefore impact the sex ratio of such species. So here we have the human sex chromosome, what the X looks like and what the Y looks like, and just showing how the XY system is as follows. A male and female will cross and how they will produce an XX female or an XY male. And the male, the sperm, is always the one that will determine the gender of the offspring. A gene located on either sex chromosome is called a sex-linked gene. The X chromosome carries many X-linked genes that control the traits unrelated to sex, and the inheritance of white eye color in the fruit fly illustrates an X-linked recessive trait. So here's a look at red eyes and white eyes, and this is showing that it's a sex-linked gene. When we cross a male and a female, we see that the red eye allele and the white eye allele is associated with one of the sex chromosomes. So that's a sex-linked trait. Most X-linked human disorders are due to recessive alleles and therefore are seen mostly in males. A male receiving a single X-linked recessive allele from his mother will have the disorder, and a female must receive the allele from both parents to be affected. So this is just a look at hemophilia, which is a blood disease in the royal family of Russia, showing how the carriers and someone who is normal and how the traits can be passed down between male and females. And then I think, so Y chromosomes can provide data about recent human evolutionary history because they're passed on intact from father to son. I think we're going to stop the chapter here because we pretty much went through all of it. I know this is a very kind of detailed chapter. Again, the pictures at the end serve as a good review for main ideas that you should know. And take a look at the YouTube video that gives you a little more explanation on the gametes and the Punnett squares of Mantel.